Dear students, after studying this module, you shall be able to know about liquid chromatography and high performance liquid chromatography, the principles of Hplc, instrumentation and their working, application of liquid chromatography and Hplc. Chromatography is a technique for separating the components or solutes of a mixture on the basis of comparative amounts of each solute dispersed between the moving fluid streams called as mobile phase and the stationary phase. The mobile phase can be either a liquid or a gas, while the stationary phase is either a solid or a liquid. Liquid chromatography. Liquid chromatography extracts the molecules in the liquid mobile phase using stationary phase, that is solid, or an immiscible liquid. Liquid chromatography is used for analytical or preparative applications. In column liquid chromatography, the mobile phase along with the solutes to be separated passes through the column and components in the mixture interrelate to varying degrees with the stationary phase. Components of interest dissolved in the mobile phase are separated based on their differing physiochemical interactions with the two phases. Chromatography can be classified on the basis of physical state of stationary mobile phase combinations. Liquid solid column chromatography, one of the most popular chromatographic technique, uses liquid mobile phase which slowly filters down through the solid stationary phase, bringing the separated components along with it. High performance liquid chromatography. High performance liquid chromatography is basically an improvised variety of column chromatography. Instead of mobile phase being permitted to drip down through the column under the force of gravity, it is forced through a column under high pressure of about 400 atmosphere, making it much faster technique. It also allows the use of much smaller particle size for column packing material, which in turn provides a larger surface area for interactions of solute with the stationary phase. This promotes enhanced separation of the constituents of a mixture. Another major development over the column chromatography is the use of highly sensitive detectors. Principle. Components of interest present in the mobile phase are separated on the basis of their differing physiochemical interactions with the stationary and mobile phases. The stationary phase is fine adsorbent solid, a solid that is able to clamp into liquid particles on its outer surface. The column usually used in the column chromatography is identical to pasture pipette. Pasture pipettes are used as columns in small scale column chromatography. The adsorbent solid, usually silica, is tightly filled into the glass tube to make these separating columns. For high performance liquid chromatography, it is known that the resolving power of the chromatographic column increases the column length and number of theoretical plates per unit length. Although there are limitations to the length of column because of the problems of peak broadening. As number of theoretical plates is associated to the surface area of the stationary phase, it follows that smaller the particle size of the stationary phase, better the resolution and greater the resistance to eluent flow. Schematic representation of the basic separation of the sample in the column is shown here in the given figure. Types of liquid chromatography methods. Liquid chromatography methods are named according to the mechanism involved. Liquid solid chromatography, also called as adsorption chromatography. The separation apparatus in LSC is based on the struggle of the constituents of the mixture sample for the active sites on a solid stationary phase such as silica gel. Liquid-liquid chromatography, termed as partition chromatography. The stationary solid surface is coated with another liquid stationary phase which is immiscible in the solvent, mobile phase. Partitioning of the sample between the two phases interrupts or retains some components more than the others to effect the partitioning. Ion exchange chromatography. Ion exchange chromatography is based on the computation of distinct ionic compounds in the sample for the active sites on the ion exchange resin or the stationary phase. Gel permeation chromatography or the exclusion chromatography. Gel permeation chromatography is a mechanical sorting of molecules on the basis of the size of the molecules in the solution. Small molecules are capable of infusion into more pores and are therefore reserved longer than the larger molecules. Normal phase chromatography. In normal phase chromatography, stationary phase is polar in nature while the mobile phase is non-polar, meaning polar analyte interacts more with the stationary phase, hence increasing its retention time inside the column. Reverse phase chromatography. In reverse phase chromatography, the stationary phase is non-polar whereas the mobile phase is polar in nature. If analyte is polar, it will interrelate less with the stationary phase, hence decreasing its retention time inside the column. Instrumentation and working. Liquid chromatography. It is shown here in the given figure where the injection site is. It passes through the packed column, materials are eluted from the column, goes into detector and one section goes into the waste, while from the detector a computer interprets it and computes it into a graphical representation. The mixture is injected at the injection port. The mobile phase carries it forward through the packed column where it separates into the components. The detector detects the separated analytes and the recorder, usually a computer, records this information. Following are the components of the liquid chromatographic technique. Firstly, the injection site. The sample or the mixture here does not have mobility, hence it requires a mobile phase to carry it forward. The injection site is where the sample mixture is introduced in the column. The mobile phase carries the sample mixture forward through the column. The packed column. The columns are commonly made from the stainless steel but thick glass. Polymers such as polyether ethyl ketone, a combination of stainless steel and glass, or an amalgamation of stainless steel and polymers are also used. Typical LC analytical columns range from 3 to 25 cm long with the diameter of 1 to 5 millimeters. The columns are commonly straight, unlike that of a gas chromatography columns. Particles used for packing columns have a characteristic diameter ranging in the range of 5 to 10 micrometers. Liquid chromatographic columns shows increase in efficiency when the diameter of the particle used for packing inside the column goes on decreasing. Detectors. It detects the constituents of the mixture that are being eluted off the chromatography column. The first effective inline liquid chromatography detector was refractive index detector and was reported by Tiselius and Classion in the year 1942. The conductivity detector was described by Martin and Randall in the year 1951. Other detectors used are UV detector, fluorescence detector, mass spectrometer and electrochemical detector. High performance liquid chromatography. The schematic representation in the figure below shows Hplc instrumentation. It consists of solvent reservoir, injector, pump, column detector and an integrator or acquisition and display system. The pump is considered as heart of this chromatography system. The sample is injected through a port in high pressure liquid carrier stream between the pump and the column. The separation takes place in the column, which varies from 3 to 30 cm in length and 3 mm in diameter. Typical flow rates fall between 1 to 2 ml per minute with pressure up to several thousand psi. The column effluent passes through a non-destructive detector where the properties such as ultraviolet absorbance, refractive index and molecular fluorescence is monitored. The signal is then amplified and recorded as a detector response versus retention time. The graph thus obtained is called chromatogram. The effluent may be discarded, recycled or saved for any further research or studies in a fraction collector which is synchronized with the detector. The components of high performance liquid chromatography include solvent reservoir. The mobile phase contents are stored in the glass reservoir. The mobile phase or solvent in the Hplc is generally a mixture of polar and non-polar liquid constituents whose respective concentrations vary depending on the composition of the sample. Pump. A pump sucks the mobile phase from the solvent reservoir and pushes it through the column and a detector. Operating pressures of up to 42,000 kPa, which is about 6,000 psi, can be generated based on the number of factors that include the dimension of the column, particle size of the stationary phase, the composition of the mobile phase and its flow rate and so on. Microprocessor controlled pumping systems have the capability of precisely delivering a mobile phase of either constant isocratic elution or varying gradient elution. Composition according to a defined program. Sample injector. The sample solution is usually introduced in the flowing mobile phase at or near the head of the column. Injection valve, which can operate at high pressure, is used for injection purposes. Automated injection systems are also in use these days. An injector for an Hplc system facilitates injection of a liquid sample within the range of 0.1 to 100 milliliter of volume with increased reproducibility and pressures up to 4,000 psi. Column. Columns are typically made up of stainless steel. Their length ranges between 50 to 300 millimeter and the internal diameters between 2 to 5 millimeters. They are packed with the stationary phase particles of size 3 to 10 micrometers. Columns are packed with solids like silica or alumina. These columns are called homogeneous columns. If stationary phase in the column is liquid, the column is termed as bonded column. Bonded columns contain liquid stationary phase which is bonded to the solid support, which is again usually silica or alumina. Detector. Since the quantity of material applied to the column is very small, it is imperative that the sensitivity of the detector system is high. Commonly used detectors are UV detector, fluorescent detector, mass spectrometer and electrochemical detectors. LC-MS is also used to develop highly precise and reproducible assays. Modern day mass spectrometers are extremely sensitive and LC-MS assays are now viable replacements for many immunoassays. LC-MS assays will possibly be most beneficial in the clinical biochemistry lab when used for the multiplexed and screening type assays. Data collection devices, chart detectors and electrogenic integrators are used to collect the signals from the detector. These chart recorders and electronic integrators vary in their intricacy and in the ability to process, store and then process the chromatographic data. The computer integrates the signal of the detector to each constituent and produces a chromatogram that is easy to read and interpret. Interpreting the output from the detector. The output will be recorded as a series of peaks, with each peak representing a compound in the mixture passing through the detector and the absorbing UV light. The retention times are used to identify the compounds that are present with the respect to pure samples. Standards of those compounds injected under similar conditions. Retention time. The total time taken by a particular compound to travel to the detector through the column is called as retention time. This time is initiated at the moment the sample is injected till the point at which the display shows a maximum peak height for the compound being run. Different compounds show diverse retention times. For a particular compound, the retention time will vary depending on the following factors: pressure applied, this affects the flow rate of the solvent; nature of the stationary phase, not only what material is it made of but also the particle size; precise composition of the solvent; temperature of the column. This means that these conditions have to be vigilantly restricted if using retention time is the method of identifying the compounds. Column efficiency and selectivity. The column selectivity depends upon the partition coefficient K, where K is given by the relation as K is a ratio of concentration of the solute in the stationary phase to the concentration of the solute in the mobile phase. The Van-Demeter equation HETP is equal to A plus beta upon micro plus C micro. A is a constant which signifies the various probable paths that can be taken by the analyte through the stationary phase. It decreases if the packing of the column is kept as small as possible. B is a constant that describes the longitudinal diffusion that occurs in the system. C is a constant that describes the rate of adsorption and desorption of the analyte on the stationary phase. A, B and C are constant for any given system with same analyte, stationary phase and mobile phase. So, the flow rate micron must be optimized accordingly. If the flow rate is too low, the longitudinal diffusion factor beta by mu will increase significantly, which will increase the plate height. At low flow rates, the analyte spends more time at the rest in the column and therefore longitudinal diffusion is a significant problem. If the flow rate is too high, the mass transfer term C mu will increase and reduce the column efficiency. At high flow rates, the adsorption of the analyte to the stationary phase results in some of the samples lagging behind, which also leads to band broadening. Applications: biochemical screening for the genetic disorders. A number of marker amino acids and the acyl carnitines are measured in the dried blood spots after the extraction and the derivatization in the microtiter plates. Screening strategies have been developed for the galactosemia, sickle cell anemia and lysosomal disorders. Therapeutic drug monitoring and toxicology. Assays have been developed for the immune suppressants such as cyclosporine, tacrolimus, sirolimus, everolimus and mycophenolic acid. Measurements of steroid hormones. Difficulties with the measurement of the low testosterone and dihydrotestosterone levels found in women and children using conventional immunoassays have prompted the development of several highly sensitive LC-MS assays capable of providing the reliable measurements in these groups. Used for the separation of the polar compounds such as drugs and their metabolites, peptides, vitamins, polyphenols and steroids. Used in the clinical and pharmaceutical work, as it is possible to apply biological fluids such as serum and urine directly to the column, preferably using a guard column. Applied to variety of natural products such as nucleic acid, carbohydrates, lipids, amino acids, bile acids and manufactured products such as pharmaceutical herbicides, pesticides, surfactants and antioxidants. Forensic application of Hplc includes the analysis of drugs, analysis of explosives, ink analysis in case of question documents, dyes in illicit drugs, fibers, lipstick smears, food and comparison of solid samples. Hplc can analyze very small samples and quantify the trace amount of impurities in the drug samples. Various drug enforcement administrations agencies uses Hplc to detect trace impurities in the drug samples. The profiles of these impurities in the drug samples can be matched to the specific method of manufacturing. Since each drug cartel has slightly different manufacturing system and location, this information can be used to determine which cartel produced a specific sample of drugs. To summarize, chromatography is a technique for separating the components or solutes of a mixture on the basis of the comparative amounts of each solute dispersed between the moving fluid streams called as mobile phase and the stationary phase. Liquid chromatography separates the molecules in the liquid mobile phase using a solid or an immiscible liquid stationary phase. High performance liquid chromatography is essentially a more efficient form of column chromatography. Instead of a solvent being allowed to drip through the column under gravity, it is forced through under very high pressures of up to 400 atmosphere. Types of liquid chromatography are liquid solid chromatography, liquid liquid chromatography, ion exchange chromatography and gel permeation chromatography. The applications of liquid and high performance liquid chromatography includes biochemical screening of genetic disorders, therapeutic drug monitoring and toxicology, measurements of vitamins and related metabolites, measurements of steroid hormones and so on.